Now, last lecture, we talked about how many of the drugs are not safe and how women were excluded from clinical trials. So as you guessed, there is some regulatory bodies that regulates the drugs manufacturing and the drug industry, uh, um, all of it. And today we will talk about this, how the regulation and how is the drug is discovered and then marketed in the market. Before we start, just a quick reminder of what are the nature of drugs that we have these days. So what are these drug modalities? So most of the drugs are called small molecule. So they have they're chemically synthesized. They have a molecular weight of around less than 100 Dalton. And most experience in drug discovery is based on the small molecules, and it has the least manufacturing complexity. So the manufacturing of these drugs are less demanding, and the storage as well, and the transportation is less demanding than the other modalities. Now, you probably heard of the word the drug versus biologics. So we know with the advancements of technology, then came the protein-based drugs. These protein-based drugs, either, for example, enzymes or antibodies, so or peptides. These peptides or enzymes, for example, they replace a missing hormone, let's say insulin or growth hormone. These are called protein-based. Okay, they are larger in size than small molecules, and their manufacturing is more difficult because you need a, a a living cell to produce these cells. For example, you need bacteria or you need yeast. Also, antibodies. We will talk about the antibodies in the treatment when we talk about the anti-cancer. A lot of antibodies now are used as a treatment for cancer. They are also called protein-based. Now, a less complex and a smaller drug entities are the nucleic acid therapeutics, which are the target of these. So most of the drugs, the target of the drugs are proteins. Okay, so they work on protein. So these nucleic acid, they are, for example, single-stranded RNA or DNA, or even double-stranded mRNA. So they consist of nucleic acid. They target for example, the messenger RNA or the DNA itself. So the function of this is to, especially in monogenic genetic diseases, to reduce, for example, the production of certain, the expression of certain protein, or even manipulate the splicing and manipulate the genetic, so the correct the for example, the splicing error, and therefore there is a gain of function of these proteins. Even more complex treatment modality is the genetic therapy and cell-based therapy. So in genetic therapy, you will have the correct gene used. The scientists put this gene in a vector, usually a virus, then they insert this into the body. And then the idea is that this virus will go into cells with the defect gene and replace it with the correct gene, okay? So it's a very complex modality. It's a kind of more recent modality as well. The cell-based treatment as well, they take the doctors will take cells from the patient themselves or even from a volunteer, and then they genetically modify these cells and then put it back into the body to fight diseases. For example, if you probably heard about the CAR T cells in cancer therapy, and where they take the T cells. The T cells are immune cells that kills foreign bodies, okay, and they can help uh, in killing cancer cells. So they take the T cells from the patient or volunteer, and then they teach these T cells to fight the cancer cells, put it back in the patient, help in fighting the cancer. And then you have the microbiome and using living, for example, bacteria to enhance the number of and function of the good bacteria that is found in the body. So these are different modalities, and many of these modalities, they will have probably different regulation, but in general, they will go the same regulatory process. Now, the FDA, probably heard about the FDA a lot. The FDA is it's a short for the Food Drug Administration, منظمة الغذاء والدواء. This is based on in America. It's a major regulatory body in the world, and like it's for the US, but it's very huge that a lot of drug companies we want to get, even if they're not American, they want to get the FDA approval. Anyways, so there are many major historical events that eventually led to the FDA that we know, and until now, the FDA regulation, it keeps changing. Its regulation depends on the new data and the new demand. So one of these, the first, for example, one of these health crises that led to the FDA, for example, in 1902, there was a diphtheria antitoxin and chicken pox vaccine that they were contaminated with tetanus. And if you see this in here, this is called Jim, this horse is called Jim. So what happens back then is that diphtheria toxins are injected into this, for example, this horse, and then the horse will the body will form antibodies against these toxins, and then the doctors will take the serum from this, the blood from this horse, and inject it into the patient in to hopefully these antibodies to fight the diphtheria. And it was working. And this, in particular, this horse called Horse Jim produced a lot of patches. And what happens is, and as well as the chicken pox vaccine, these animals were carrying tetanus, and they were not tested for tetanus before distribution of the drug, and so many people died. Okay, so so people said, okay, we have to have a certain authority and to give license to these drug manufacturing, and also they give this for the public health services, the authority also to inspect the manufacturing process. So this was let's say the infancy of FDA. It's still not called FDA, and it was like, but it eventually led to the FDA that we know. The second crisis was in 1906, where there was a public outcry about complaining about the condition of the meat packing industry. They were unsanitary, they were dangerous. So the public health authority responded by releasing this the Pure Food and Drug Act Act, and what happened is these authorities prohibited these companies from misbranding the food and drugs. So even the label start people start to control them. Now, in 1938, this is where, let's say, the FDA was called FDA. It was after the strep elixir tragedy. This tragedy. What happens there? There was a sulfonamide. It was an antibiotic. And this one company, Massengill company, they wanted to produce syrup from these antibiotic. Okay, so for the mostly to target children. And what happened? This chemist found this amazing compound, was where this compound was perfect, diethyl diethylene glycol. It has no taste, it has no smell. It made a good solvent, so the drug could be manufactured as syrup, زي شراب يعني. However, and then they did not test for safety, and then they distribute the drug, and many people, most of them children, more than 200 people were killed by this drug because it was toxic. This diethylene glycol is now used as it's an antifreeze. It's very toxic drug, and this where the Food and Drug was then called Cosmetic Act established, and this said, okay, this gave authority for this uh, regulatory body to that all manufacturing should seek approval from the FDA before marketing any new drug. Now, as we said, the laws continues to evolve depending on what's happening. And you remember from, if you heard the last lecture, the thalidomide tragedy, where this drug was marketed into pregnant women for to fight insomnia and morning sickness. However, it was very teratogenic, and many miscarriages and many developmental error occurred. Okay. So after this tragedy, the FDA required a proof of efficacy, safety, also report of adverse event, and also patient must provide informed consent to participate in clinical trials. And as I said, still evolving, and you remember the act where they changed the pregnant woman from vulnerable to non-vulnerable, or the act, or in 1993, where the FDA said that, okay, women should be involved in clinical trials. And I just wanted to add this because this act, the FDA Modernization Act, it was issued in 2022. It will change a lot of things. So we will talk about the developmental process, and the animal testing was a cornerstone of drug development. It was needed, especially to establish toxicity and efficacy before going and trying the new drug into humans. So what happens is that we will talk about it, that a lot of these animal studies, they did not replicate what's happening in humans. So a lot of drugs failed, and most of the failure is because of toxicity and and there is no efficacy. So what happened is, with the advancement of the technology and the in vitro, especially in vitro يعني خارج الجسم باللاب يعني, there are developed, the scientists developed these amazing in vitro studies that can recapitulate what's happening in the human body. And what's happening is that the FDA is trying to phase out the animal studies and replace it with these more advanced in vitro studies. These include this advanced cell-based assay, where they you have different cells in one dish, and so if you imagine an organ is composed of many different cells, it's not one cell. Okay, so these a lot of these advanced cell assays try to recapitulate this and have more than one type of cell. Also, organoid. These are organoids are these. It's a technology where they can grow a mini organs, يعني a mini organ mimics. It's not the actual organ, but it's look like a lot. If you can see, this is a brain organized with many mini brain-like tissues in, for example, petri dish, which will represent to a large extent the human brain. Okay, another technology evolved is the organ on chips. Now it's more and more evolving. This is the chip where there is these wells will have different type of cells representing an organ, and between them are connected with these microchannels with they call it microfluidic technology. Okay, um, so all of these are trying to replace human animal study by mimicking the organ itself, and it's been for some of them been useful in detecting toxicity, for example, in these before going to clinical studies, and of course more recently using advanced artificial intelligence as well could help reduce animal study before going into clinical trials. Now, just one note, we say FDA, this is more based on America, in the equivalent in Europe called EMA, the European Medicine Agency. So if you heard FDA, AMA, both are regulatory, drug regulatory bodies. Okay, so here we start about talking about the stages of drug development, where you start with drug discovery. So drug discovery requires discovering the target, so what on what these drugs are going to work on, and then discovering the compound themselves that are the medicine itself before going to preclinical, where the preclinical is in nowadays is more it's the animal studies where we said before it's as well as more recently more advanced in vitro studies before going to first time in human study, which is the clinical studies. So we have the drug discovery, the preclinical, and the developmental or clinical developmental phase. We will start. Okay, this is a nice graph. I found this. So drug discovery and development, it's a very expensive process, and it takes traditional one takes a lot of time. So this is just a nice representation. So let's say before preclinical studies, the only 85% من ال discovered compound, developed compound will enter the preclinical studies. And then in the preclinical studies, after this, only 70% will go into human studies. So phase 1 is the first human trial. And in this phase 1, you can see here the success rate is only like half of them will move into phase 2. And in phase 2, this is the valley of death. This is where most drug program ends, because almost 2/3 of the drug fail phase 2, so they don't advance into phase 3. When these drugs advance into phase 3, there is a chance of 30% chance the drug will not go into the market. And even if the drug is submitted and went into the market, there is still 10% chance that the drug fails in the market. Okay, so if you want to take all this in one in numbers, so almost 90% of the drugs will fail clinical trial. It's huge money and huge number of drugs. If a drug company want to develop a new drug, it might cost up to $2 billion before going into the market and starting have يعني الدرug company start take المصاري تبعتها يعني. And it's almost the drug discovery, it's very long. It may take from يعني it will differ from research to research, but it may take up to 10 and 15 years to discover to go into the market. So it's a long, expensive, and highly risky industry because there is a high attrition rate. High attrition rate يعني الfailure rate is عالي كثير بالdrug discovery. So we said the first thing is to find a target where the drug work on, or pathways that the drug will work on, and this is called target validation. And this usually probably in the academia setting. So many of the researchers in academia and professor in the universities will investigate these pathways and these targets and try to link it to a disease. And target validation means you want to see, for example, let's say you have an enzyme, you have a protease. The scientists found that it was upregulated. There's a lot of this protease in a certain lung disease. Okay, so they think, if we can inhibit this protease in this certain lung disease, the theory is probably there is going to be beneficial effect for the patient. So they need to test this as well in vitro and in vivo studies to establish this validation. Okay, so it's also a long, and many projects will work on target validation. And one of the things that is linked to the idea of target validation is the strategies to identify drug starting point. So there are two strategies to identify, what the drug, or like what should I do, how to start a drug development program. The first one is called the phenotypic or compound-centered approach. It's an older still used. It's an older method where what they do is that they screen a lot of compound in cell model or even an organ model, and then they will see the effect. So they know the compound, but they don't know the target. They don't know the target or the pathways. And so the mechanism so they see, for example, they give this compound and then the tumor shrink. There was a tumor shrinkage, or the tumor stopped metastasizing. So all of this, you see, this is the phenotype. Phenotype يعني العرض yeah, probably genetic, what's a phenotype, okay? So they see this effect, but they don't know exactly the mechanism. Sometimes they go back and try to investigate what's happening and the mechanism. So the first time they see the drug, the drug, and they know the effect, but they don't know the mechanism. Now, the problem with this approach, if you don't know the mechanism, you probably will not predict the toxicity or even the efficacy accurately. Okay, one of example is the natural product, which is an anti-cancer product. We're going to talk about it in the anti-cancer therapy is paclitaxel. Okay. The other approach is that no, the target is validated. They know the target. Okay, let's see, it's a protease. They know cathepsin. It's a protease. They know their target, and then they try to develop a compound that can modulate the function of this target. In this situation, you know the mechanism of action, for example, inhibition of the enzyme or potentiation of the potentiation of the I don't know, the ion channel or whatever. So here it's easier to predict the toxicity and the efficacy. Okay, so here the target is first validated, and then you look for a compound that will manipulate and the action of the target. Okay, so the first stage is after, let's say, we have a validated target, is the drug discovery itself. And when we talk about drug discovery, these stages are more for, let's say, the target-based drug discovery, and also more about the small molecule drug discovery. However, it has some similarities to other drug modality. So the first thing is hit identification. Hit is that you hit it. انت أصبته. يعني you found something that is active. Okay, you find something that's active. So what's happened is that the drug companies will have these large, we call it libraries of compounds, and these screen thousands to millions of these compounds. And assays they probably develop. For example, they want to test lung diseases or an anti-inflammatory effect. So they will develop assays to measure the anti-inflammatory effect of these hits. And then they, from these thousands to millions, they will probably identify a couple of hundred. Okay, so hits there are identified as starting point. They have activity. They could manipulate the activity or of this certain target. So however, these hits are very weak binder. At this stage, we're not looking for molecule with high potency. They have a low potency. They have weak activity, and they have what we call a drug-like characteristic. So a drug-like characteristic has to do with, for example, solubility, toxicity. So many of these were probably very toxic, or they don't have the appropriate solubility for a drug to be, for example, absorbed, and the ADME effect of the drug. So the next stage is hit to lead characterization. In this stage, the scientists explore the hits chemistry and biology and prioritize those ones that can be developed more. Okay. So I'm talking very fast. You don't have know these details, as just know these stages, and it's just to appreciate how complex it is. Okay, the drug discovery itself. So after this, the goal is to identify a lead series with improved potency and selectivity and some initial ADME properties like improved solubility, maybe improved impermeability, and maybe the metabolic stability. So they identify a couple of compounds, probably with a different chemical entities. So always the drug companies like to have a backup. So something failed, they have a backup to go for. Now, after the identification of these lead series, it comes the lead optimization stage, where here the goal is to have a compound or a couple of compounds that will go into preclinical studies and hopefully into clinical studies. And these here, a more extensive, probably animal work and ADME screening and toxicity screening will these compounds will be tested against these more screens to try and identify a clinical lead. Okay, now let's say we have a clinical or preclinical candidate, and a couple of these candidates, let's say maybe at this stage, maybe you'll have 10 going to animal work, or the preclinical stage. So at this stage, now it's historically and until now, it's the preclinical studies are majorly done in animal models, and the and the and the and the goal of the preclinical development is you need to test these compound. So when the drug company want to file to the FDA that they want to try it to in human, they have to have all these files and these data that it is majorly it is safe and not toxic and effective and probably effective in a human. So the work falls into four main categories: safety, pharmacology, to check if the drug does not produce harmful effects, for example, cardiotoxicity, for example, or it's not, or hepatotoxicity. And then they go to also they need to show in animal studies there's no genotoxicity, and they need to determine the maximum non-toxic dose for the drug. Okay, every drug has a toxic dose, a maximum toxic dose, or so they want to see how much you can give to the animal, and probably and eventually how much of the drug dose you can give to the human before it's becoming toxic. Also, you have probably in the same animal model of toxicology model, you have the pharmacokinetic and pharmacodynamic study, where you need to establish efficacy. Okay, so this drugs reduce the inflammation, for example, in the animal studies or not. The drug company need to show or need to have these pharmacodynamic data about bioavailability, whether the food is affecting the drug absorption, elimination, metabolism of the drug. All of this, the drug company need to show for the FDA, and also it will help the program to design the dosage of in the clinical trial. One important thing is as well is the chemical and pharmaceutical development. So in parallel to drug discovery, the active so when you take a drug, it's not only the active ingredient. It's not only, for example, this enzyme inhibitor. There is a lot of additives in the drug itself. Okay, in the tablet you take, for example, this is what we call it helps the delivery and stability of the drug, and this now comes here's what's called by chemical and pharmaceutical development. And one thing as well, all of these chemical reaction that is used in discovering the drug, probably it will not be appropriate for a mass production. So the mass production, it's another field of research. So you need, we know this compound. Okay, we synthesize this compound following this chemistry, but in many cases, this chemistry is not feasible. It's probably expensive, explosive, so it's not very good for the environment. So this is what's the chemical feasibility of large-scale synthesis means. Can the drug company make a lot of this drug, because it's now you need to distribute it to many people, right? In phase 1 or phase 2, or eventually to in the market. And also assess stability of the developed formulation. Is it stable? Can you store it? How much is the shelf half-life of the drug? All of these need to be assessed and admitted to the FDA before going into a first in human trial, which is the phase 1. So the second phase is finally you get to test the drug in humans. So before the drug company can submit, can design and test the drug into human, they need to submit something called IND. So the investigational new drug submitted to the FDA. The FDA review all the toxicology and preclinical studies, and then may or may not give the drug company the okay to investigate in human. Okay, there are four distinct phases from 1 to 4. The first three, one is it's only at trials, but however the 4, it's post-marketing. So the drug now can be distributed, and the drug company can sell their drug, but still need to have studies of safety and efficacy in the public. We will talk about each of these phases afterward. But before that, the protocol of these trials differ from one to another, and you hear a lot of these. Probably would hear a lot of these randomization, controlled, or a blinding. So what is a randomized clinical trials? So a randomized clinical trials is you probably in the clinical trial, you will have a group that will take the treatment, and the another group will not take the drug. Okay, so this is the control. So randomization is every participant will be assigned to the different arms or the different groups by chance. So the doctor does not decide, the patient does not decide if he wants to take the treatment or not. So this helps avoid something called bias. Bias, I think بالعربي زي الانحياز يعني. To understand this, for example, to ensure that people of the same sex or age, for example, are not all assigned to the same treatment group. Okay, so you want to compare, for example, different treatment. So if this treatment X gets all the women, for example, it will not be very validated, or the data will have some bias error. If it have only, for example, this treatment will have only women in it, or this will treatment more older people in this treatment group. So to avoid this, randomization, assigning people by chance, may reduce this. Okay. The other thing is you hear is the controlled clinical trial. It's when you have a so when you have a data, you want to compare the new drug to something else. So you give a group the treatment or the new drug, and the other group you don't give them, and then you compare, is it effective, is it toxic or not? Okay, so to give more comparison. So in general, participants are put into a study group. This group, they receive the new medicine, or a control group. The control group, they could receive something called placebo. Now, placebo is this sugar pill. Okay, so it's let's say they're trying to give the clinical trial is exploring a new medication in a form of a capsule. Okay, so they give the study group this capsule. Inside of it, it's the drug. However, if the trial uses a placebo, the comparator group will take the capsule, but it will not have the drug. It will have something else that look like a drug, but it's not, the active ingredient is absent. So this is called placebo. Or maybe the comparator will have only received the current standard of treatment. For example, you want to try this drug on asthmatic patient. So you will have asthmatic patient, and then with the new treatment, an asthmatic patient, you cannot stop treatment of asthmatic patients. They need to have their current treatment. So this is what the current standard treatment means. Or even they don't have the control group will have no treatment if it's feasible. So it's the aim of this is to isolate effect of the new treatment and to better establish a causal relationship. So you can say, okay, probably this drug caused this effect, whether efficacy or toxicity. Also blinding. You hear these word, okay, this clinical trial could be single blinded or a double blinded. So in a single blinded trial, participant do not know whether they're receiving a study medicine or a placebo, but a researcher know. In the double blind, which is more, let's say, probably will give more accurate and valid result, is where neither of the participant or researchers know whether the participant are getting the study medicine or placebo. So the aim is to prevent the bias. So if let's say the participant know they are receiving, for example, the active ingredient, this will probably uh, affect them psychologically and then affect their assessment of the drugs. So any probably they will feel, oh, okay, improved. Actually, they're not improved. They're just feeling improved because they know they're taking a new drug. Another example is, for example, if the participant does not know that, okay, it's only placebo, it's not going to work. So they will not probably adhere to the protocol or they adhere to the treatment or the assessment of the intervention. If they, for example, the research, what's the benefit of blinding the researcher? So if they, for example, I'm a doctor and I know this person is taking the drug in test, and this patient is not taking the drug in test, so probably this will give more reluctant. The doctor will be more careful in assessing the one who's taking the new medication versus who's not taking the new medication. So the attitude and attention of the treatment will probably differ if the researcher knows. Okay, sometimes there is something called emergency unblinding. That means the doctor and the participant need to know whether the patient is taking the drug or not. This happens in research where, for example, a very toxic, they discovered, for example, in phase 2 or phase 3, that the drug is cause a severe side effect. So the researcher, the doctor need to know who took the medicine or not, and as well as the patient. They need to know they are at risk of this side effect or not. So this is called emergency unblinding of the clinical trials. Here, just a representation of a randomized control trial. And remember that randomized control trial is the gold standard in evidence-based medicine. Okay, they give the most valid result. Okay, so before going and talk about the different phases of clinical trial, just to mention here, there is something. These are the regulation also of the clinical trials. Try to ensure the safety of the patient. So it should be there should be some ethical consideration. The drug company need to follow some ethical laws to make sure the patient is not put in, or the volunteer or the participant are not put in an extra danger. So these laws are established by the International Conference on Harmonization and Declaration of Helsinki. And the four major ethical principle is: first, the trial must minimize the risk of participant. So, for example, they monitor the volunteers. They should monitor the volunteers very carefully, and it's the drug must be, for example, given in a clinical setting. Not okay, you take the medicine and go. No, it should be probably taken in a health center to monitor any side effect. The provision must be made for the overall care of the patient. Yeah, the same similar to the you have to care about the patient. So in some trials, when you use patients, you need to give them their if for example they're not taking their the new drug, you need to make sure they are taking the standard care of medication, right? And the third point is the investigator is responsible for terminating the trial when the risk become incompatible with the goal of the trial. So if there is associated okay, so they are in the third phase, okay? The second, and then a lot of patients suffer from cardiotoxicity, for example. A lot of them died from, and even if it's not a lot, sometimes it's some percentage of incidence of death, then they need to stop the treatment, okay? Adverse event must be reported immediately to an ethics or safety committee. Let's make sure the drug company is not hiding anything, just like what happened with the thalidomide. And every adverse effect, every side effect, whether toxic or not toxic, need to be administered. So clinical trials, we start with the clinical trial. This is the first time this drug is used in human. It's more for safety and toxicology studies. So 10, 20 to 100 subjects are volunteers to participate in a clinical trial phase 1. They're most of the time they are healthy, normal volunteers, but sometimes, especially if, for example, the drug has a high level of toxicity, for example, cytotoxic drugs in cancer therapy, patient can be used. Okay, the duration, if it takes weeks to several months, and in this trial, the participants are closely monitored in health centers, and this is why it's also expensive. The clinical trial is expensive because you also monitor the participant very closely. This takes a lot of doctors, a lot of tests, which are not cheap. So the purpose is mainly to check for potential harm effects, for example, cardiovascular, respiratory, or hepatic or renal function, and also to determine the DLT or dose-limiting toxicity. Okay, from this, you can determine the dose-limiting toxicity or confirm it, because they will have some idea of what is the dose-limiting toxicity in animal, and you need to confirm it with it. Try to confirm it in human. Now check for tolerability. Tolerability is different than toxicity. So tolerability maybe it's the unpleasant symptoms, for example, headache, nausea, drowsiness, and determine the maximum tolerated dose. Okay. Also, can be used, the data from clinical trial phase 1 can be used to explore the pharmacokinetic, whether, for example, does food interfere with the oral bioavailability, the secretion, the metabolism. They can draw blood and see what's happening on the what's happening to the drug inside the human. So explore the PK properties. Also, sometimes there is something called they will try to assess the pharmacodynamics. The pharmacodynamic is what drug does to the body, okay? And this is called sometimes proof of concept. For example, it's not always done or feasible, but for example, does a novel analgesic block experimentally induce pain? So you bring healthy volunteer, you induce pain in these healthy volunteers in a safe way, okay? And then you give your analgesic, اللي هو pain reliever. And then you see, does it alleviate the pain or not? Okay, or for example, in some cases, you can induce inflammation inside the, for example, in the the lungs of the patient. They call it lipopolysaccharide challenge. So you take healthy, you induce inflammation. This is very closely and it's safe. It's not that dangerous. And then you try the anti-inflammatory drug, and you check does it block the anti-inflammatory drug or not. And this is what it's called the pharmacodynamic assessment. Okay. In some cases, sometimes, and I think more and more drug company is adapting this, is called the therapeutic or microdosing trials, or phase 0 trials. In these, a couple of maybe 5 healthy volunteers, they will be taking the drug in a subtherapeutic. It's not gonna it's subtherapeutic. It's less than. It's not gonna have an effect. Okay. Um, this is to see, and then they give this drug, and this drug probably will be tagged with a radio label, and then they give it to the patient and see by imaging or biopsy where did the medicine go. So what happens is what's information you take from this is, does the medicine go, for example, to the target tissue? Does it or not? So it will kind of predict efficacy or not. Okay, because sometimes, okay, in vitro, in vivo, in the animal studies, okay, it's you see the drug is effective, but in many times, this drug inhibit this enzyme. Okay, but many times in clinical trial there's no efficacy. So the efficacy problem could be as a result if that the drug is not reaching the target. So if you want this drug to go to, for example, the brain, and it's not reaching the brain, it will not help the alleviate the condition. So this is what's the purpose of this. Does the drug engage to its target? Does it go to its target organ? So they call it the phase 0 trials. Now, phase 1 trials are dose escalation trials. So what happens? So يعني dose escalation يعني أنت بتزيد الجرعة شوي شوي. And you can guess why. يعني أنت you try this drug. أول مرة you try it on human. So you want to be this as safe as possible, right? So you need to increase, our researcher need to increase the dose gradually, not give them a huge dose from the beginning. No, they need to, and this is called dose escalation. And phase 1 usually falls into phase 1A and phase 1B study. So in phase 1A, it's called a single ascending dose. So they start with a low dose level of this low level single dose. So you have a group of patients. Let's say 5 patients. You give them this low dose. Okay, you see is there a serious effect? No. Then another group, not the same group, another group, you will give them a higher dose, and you keep this switch, يعني moving from one group to another to and give them a higher dose until the maximum tolerated dose is reached، أو إنه هم الجروب نفسهم حددوا okay, this must be the highest level of safety. Okay, now this is phase 1. Okay, so you determine the maximum tolerated dose in those patients in those different groups. Now in phase 1B, the second phase is phase 1B, which is called multiple ascending dose. So several groups each receive a different repeated dose. So this, let's say, you give this group first this, let's say, ما بعرف 200 200 جرام. بعدين you give them مثلا ال 200 جرام بعد مثلا 4 ساعات أو بعد مثلا 6 ساعات، وتشوف if there is يعني عشان to determine the best dosing، يعني كل قديش تعطيهم، كل 3 ساعات، كل 4 ساعات، مرة باليوم. ف this help so the same group will be given the repeated doses. Um, Now, all of these study, as we said here, and for example, here, all of these studies is to help establish the dose regime for phase two. All of these escalating dose escalation study is to help the dosage for help the design of okay, I'm not sure what happened. Hopefully, يعني recording still something wrong happened. Sorry for the pause. Okay, so we reached the phase two study, which is the efficacy trials. Here, up to several more patients involved. Subject here are patients, not healthy volunteers. Result of phase two will help evaluate whether to proceed to phase three or not, because you are يعني establishing efficacy and also يعني you still evaluate ال toxicity. It's also pinpoint additional data that must be collected in phase three. For example, does the drug need monitoring liver function or not? And inform the appropriate sample size of phase three. So the objective is to determine effectiveness and to continue monitor safety. And in many times phase 2, and this is why phase 2 يعني معظم الأدوية تفشل بphase 2، لأنه أنت you go from 10 ل 100 maximum patient ل several hundred ل thousand. So أنت you include more people. When you include more people, when the sample size increases, that means these side effects you can detect it. يعني you have a better chance to detect the side effect with the more rare side effect. Okay. They also evaluate dose response relationship and dosing regime and finalize or enhance the dosing regime, establish optimum dose and frequency of administration for phase three. Now, phase three is the large-scale testing. Here you have 100 to 1,000 patients conducted usually at multiple sites, maybe different geography. A lot of مثلا بأمريكا، بأوروبا، بدبي، مثلا centers in Singapore. So here you have multiple geographic. This means you have different ethnicity, and probably the efficacy and the toxicity in different ethnicity probably will differ. Okay. It's very costly and difficult to organize, as you can يعني imagine. The goal is to establish and confer the treatment therapeutic benefits and identify any long-term side effect. لأنه usually, especially for chronic diseases, these studies may يعني يضلهم سنوات، سنتين، ثلاث. Okay. بعد ما if successful, if phase 3 successful, the drug company will submit an NDA, new drug application لل FDA, which may take up to 1 year to 2 years حتى ال FDA review هذول كل الداتا هذول وإذا يعطيهم ولا لا. Again there is يعني 10% chance they will not get this NDA. Uh, so from يعني بس هيك just a number حكينا قبل إنه يعني 8% of compounds entering phase 1 will eventually be approved. Only 8%. يعني تصوروا عدد الكمية الهائل من ال failure rate. Okay, so the drug is in the market. ال FDA أعطت ال approval بس still ال FDA require من ال drug company إنه still okay make more the studies, make more يعني surveillance on called post marketing surveillance على ال drug. Here you give it to the drug and many people will take a different ethnicity, different gender, people will have probably ages مختلفة, polypharmacies, they will take different drugs. Here is the real test again. It's in the real world setting with a really large number. So you need to continue monitor the new drugs. And many of them pharmacovigilance they call it. So انت كدكتور إذا مثلا البيشنت أجالك وقال لك حكى لك أنا okay I tried this drug, it's a relatively new drug and I start to experience this and this and this, you can report these symptoms in في بسموهم pharmacovigilance يعني bodies أو يعني موجودين online. يعني ال doctors now might have these are the databases where a doctor or patient can report any adverse effect. Okay, as well as حتى ال drug company نفسها they will have kind of trials اللي هم أو studies mostly observational studies حتى يشوفوا whether there is toxicity أو efficacy of these drugs in real time حتى after إنهم بلشوا يعني ياخذوا profit, بلشوا يبيعوا الدرug. Okay. Sorry. So the objective is conduct ongoing safety surveillance, identify rare adverse reaction. حكينا أنت بتستخدم لفترة أطول, patient samples أكثر, assess efficacy again, and sometimes optimize the drugs use. Sometimes شو بصير إنه okay, it's useful for this patient but not useful for this. So this is what it means by optimizing the drug use. So phase 4 may result in يا إما خلاص أوكي ينجح ويصير ناجح أو إنه it's as it's intended or as expected أو it may use in limiting the use of a drug to a particular patient group. For example, for quinolone, they discovered it's very toxic and it's limited, for example, to very severe infection. Or even it can lead to withdrawal of the drug. هون الخسارة يعني هون عن جد خسارة البلايين. فإتس the withdrawal of the drug, for example, rofecoxib, it's a non-steroidal anti-inflammatory painkiller. كانوا يحكوا إنه هم وصفوه للarthritis pain. However, it showed negative cardiac side effects, so the company needed to withdraw the drug. خلص سحبته من السوق. So this is summary of the also summary of drug development process here: drug discovery, preclinical development, then clinical development, when to submit for regulatory approval, and phase 4 اللي هو post marketing surveillance. Just a quick هنا بس إنه drug repurposing. So drug repurposing شو يعني؟ أنا عندي drug، أوكي، هذا ممكن أنتوا سمعتوه كثير أيام الكوفيد. أنا ما بعرف شو قديش بتتذكروا من الكوفيد، بس كثير بالكوفيد أيام الكوفيد كنا نسمع drug repurposing. شو يعني؟ أنا عندي drug، هذا الدرج مثلا يستخدم للهيباتيتس أو يستخدم للاتش اي في. I want to test it مثلا للكوفيد. أنا عندي موجود، أنا عندي السيفتي تبعته موجودة، فأنا I need to see if it's effective مثلا بالكوفيد. So you have the drug, so you skip the drug discovery with target validation with preclinical studies probably, and you test it دغري على patient. So you skip time, you skip a lot of you save time, you save money. However, still, if مثلا the drug company want to brand this drug for a new and they want to brand their hepatitis drug, COVID, as a COVID drug, they need to go through FDA again and some testing. So still need some clinical trials to approve, especially to make sure there is no toxicity as well as no side effect in these people, and no an efficacy, and most important, is it effective or not? So this is what's called by drug repurposing. Okay, this is a quick يعني slide. It's artificial intelligence and drug discovery. I want to just to appreciate, because we are in the new era, and I'm sure things are happening so fast. Now we're talking about no animal study, reduce animal study. When يعني قبل كان الانيمال studies, you have to do animal studies more than different species, you have to file this big file about هاي. وهسه شوي شوي انه okay, we have these alternative, as well as ال AI is coming, يعني is coming even in drug discovery. And I want to talk about Rintosertib. Now, Rintosertib, I think الكومباني هو اسمهم in silico medicine. So what happened is that this in silico medicine, they developed advanced artificial intelligence called Pandaomics. So what happens is this Pandaomics, using AI, they validated a target for a pulmonary disease called idiopathic pulmonary disease, idiopathic fibrosis, IPF. And from this result, from their AI, they validated the TNEC kinase. It's a protein kinase, so it's validated this target. Then also using another advanced deep machine learning called Chemistry 42. It's an AI tool. And from this tool, they could discover a drug and design a molecule to inhibit this kinase, the TNEC kinase. So what happens is that they really accelerated the process of drug development, and actually it worked. They of course did some kind of testing, but it was faster. Okay, so it went to the preclinic and it worked, and then it went to phase 1 clinical trial. Also, it worked, and now it's in phase 2 for idiopathic pulmonary fibrosis. So here is, in a nutshell, both the biological target and the therapeutic اللي هو الchemical compound where AI discovered. هذا اللي هو اللي what make their program special. And from start to phase 1 on under 30 months. And this is a huge advancement and acceleration of the drug development. So AI is coming and it's accelerating the drug development, and it's changing the traditional drug development pipeline. Another final slide is just a couple of يعني مفاهيم ومصطلحات ممكن تسمعوها اللي هي الباتنت. So الباتنت هي إنه وثيقة إنه هاي الدرug company granted a legal right to prevent others from making the same drug. Okay, from using it or selling it. فهي يعني زي تحمي حق اللي ما احنا حكينا. يعني it's ال drug discovery is very expensive. It may reach 2 billion. ف ك development discovery. فال drug company can file for patents and تمنع ال drug companies الثانية من انهم يصنعوا هذا الدواء. عشان لما يصير له يعني submission ويصير marketing تاخذ هي اللي profit لحالها. So typically this patent is around 20 years. يعني typically it's 20 years, and many times if you go back شوي. Let's see here. When drug company, for example, around here, on preclinical studies, they will file الباتنت ويصير العد ل20 سنة. So they will lose a couple of years. So actually يعني in many times الباتنسي يعني بيستفيدوا منها من 7 إلى 12 سنة بعد ما يحطوا الدواء بالسوق يعني. This is what's patent. So شو بصير لما تخلص البيتنت؟ لما تخلص البيتنت وقبل ما تخلص بشوي مثلا another drug company اللي بيسووا شيء اسمه generic drugs. So what happens is that these drugs, these companies, okay, they know هذا والله drug بعد 3-4 سنوات رح يخلص البيتنسي تبعته. they start their own يعني شوي research. هسه بعرفوا هم الactive ingredient where it is، بس لازم يغيروا. حكينا بهاي الأشياء الاديتيف اللي لازم تنحط. فafter ما تخلص البيتنسي they will يعني هم يصنعوا هذا الدواء، هذول الجينيريك دراجز او الكومبانيز اللي بتصنع الجينيريك دراجز بيصنعوا الادويه هاي وبيطلعوها للسوق وبيبيعوها. طبعا هون صار عندنا كومبيتيشن، يعني بطل البراند نيم دراج هو الوحيد الموجود بالسوق واللي هو كثير غالي، لا صار عندنا كومبيتيشن ثانيه، لانه الاكتيف ingredient should be the same. يعني أنا a company that evolved this generic drug. الactive ingredient نفسها تبعت البراند نيم، بس أنا غيرت شوي بهاي الأديتيفز، وبعدين بروح على ال FDA make sure it's the same dose, the same يعني the same compound active ingredient. Then it will give FDA give approval for generic drug. So the difference between active ingredient حكينا there's no difference, it's the same. The price, the brand name is of course higher in cost. Generic drugs is lower because they did not go into all these long process of discovery and development. Mostly insurance they will يعني they will cover الجينيريك drug, okay, rather than brand name لأنه أرخص. Inactive ingredient they differ from that in the inactive ingredient حكينا هي الأشياء الأديتيفز اللي بحطوها، مو الactive ingredient may different from من البراند نيم. The strength with those it has to be the same. It has to be the same عشان efficacy تضلها the same. Look البراند name there is a standard in size و color packaging etc. However بالgeneric packaging and the drug itself may look different. Okay, الباكج نفسه لازم يكون مختلف عن البراند نيم. And that's us. وإذا كان عندكم أي سؤال يعني please don't hesitate and write it على التيمز.